For this lecture, we will cover Chapter 3, Antigens and Antibodies. To start off, there are some terms that you should be familiar with. Those terms are listed here, so review both of these slides just to kind of be familiar with these terms because we will refer to them throughout this lecture as well as the continuation of this course. If you have any questions on any of these terms, be sure to look them up and dive a little bit deeper into them. Otherwise, we will move on to the actual lecture content. So an immune response is actually triggered by immunogens. Immunogens are macromolecules that are capable of triggering an adaptive immune response by actually inducing the formation of antibodies or synthesized T cells in a patient that's immune system is actually working properly. Immunogens can specifically react with corresponding antibodies or synthesized T cells. Now, an antigen is any substance that may be specifically bound by an antibody molecule or a T lymphocyte receptor. Although all antigens are recognized by specific lymphocytes or by antibodies, only some are actually capable of activating some sort of antibody formation. Some may not actually be able to invoke any sort of immune response initially. For example, if there's a low molecular weight particle like a hapten, it can bind to an antibody, but it must be attached to an actual macromolecule as a carrier in order to stimulate some sort of immune response. Now, in reality, all immunogens are antigens, but not all antigens are immunogens. Cellular antigens of importance to immunologists actually include the histocompatibility complex groups and human leukocyte antigens, or your HLAs, your autoantigens, and your blood group antigens. Now, the normal immune system will actually respond to foreign particles or foreign substances by actually producing antibodies. And so for this reason, microbial antigens are also important to immunologists as far as studying the immunological manifestations of various infectious disease. And also I want to note that some of the major histocompatibility complex antigens are more potent than others in actually provoking an immune response. Nucleated cells such as leukocytes and tissues have many cell surface protein antigens that readily provoke an immune response if transferred into a genetically different or an allogenic individual of the same species. As previously mentioned, some of these antigens, which constitute the major histocompatibility complex, are more potent than others in actually provoking an immune response. The major histocompatibility complex is referred to as the human leukocyte antigen system within humans because its gene products are originally identified on the white blood cells. The HLA system is of primary importance in the hematopoietic stem cell transplants. So these antigens are second only to the ABO antigens in actually influencing the survival and graft rejection of any sort of transplanted organ. And so there is a strong association between individual HLAs and the actual immunologic disorder. Sometimes you will actually have to use HLA matched platelets when you have a patient who is refractory towards the treatment of just using a random donor platelet. In paternity testing, HLA testing is used along with a few other key points that they look at when they're doing paternity testing. It's also useful in other areas like forensic medicine, anthropology, basic research in immunology. It's also been used as diagnostics and as a diagnostic and genetic counseling tool. The major histocompatibility complex is actually divided into 4 major regions, D, B, C, and A. I'm not expecting you to know a lot of detail about these, but just know that these various gene regions and locations is what they actually use to aid in this testing. There are 2 classes of HLAs. Class 1 regulates interactions between cytolytic T cells and the target cells, and Class 2 restricts the activity of regulatory T cells. Now, the recognition system that can actually recognize and destroy the non-self material must also have safeguards in place to prevent damage to self-antigens. So the body's immune system will usually exercise a tolerance to self-antigens, but there are some situations where antibodies may actually be produced to normal self-antigens or normal tissue within one's body. So the failure to recognize self-antigens can result in autoantibodies that are directed at hormones. So an example of this would actually be like thyroglobulin, where you start seeing issues with the thyroid. Now, blood group antigens are actually widely distributed throughout the tissue, body, cells, and body fluids. But if there is a foreign red blood cell antigen that is actually introduced to the host, then you can have a result of something like a transfusion reaction, or you can see within mother baby a hemolytic disease of the fetus and newborn. So now let's look at the nature of antigens and first discuss the chemical nature of antigens. So antigens are usually large organic molecules that are proteins or large polysaccharides, and less commonly lipids. So antigens, especially cell surface or membrane-bound antigens, can be composed of a combination of biochemical classes. So an example of this would be like glycoproteins or glycolipids. For example, histocompatibility HLAs are glycoprotein in nature and are found on the surface membranes of the nucleated cells within the body, and they're composed of solid tissue and most circulating blood cells. And this would include things like granulocytes, lymphocytes, thrombocytes. Now, the response to immunizations can actually be enhanced by a number of agents, and these are collectively called adjuvants. And so an adjuvant is a substance distinct from an antigen that actually enhances the T cell activation by promoting the accumulation of the antigen-presenting cells at a site of antigen exposure, and by actually enhancing that expression of the costomodulators and the cytokines by that antigen-presenting cell. An adjuvant that is actually used in human vaccines is alum. And alum is just a double sulfate formed of aluminum and then some other element, usually something like iron, chromium, magnesium sulfate, and they're all combined with the sulfates and various metals in that vaccine. Now, discussing the physical nature of antigens, some important factors in the effective functioning of antigens include the foreignness, the degradability, the molecular weight, structural stability, and the complexity. Now, the foreignness is just the degree to which the antigenic determinants are actually recognized as non-self by the individual's immune system. The immunogenicity of the molecule will actually depend on its degree of the foreignness, in addition to the dose of it upon exposure, the route in which it is exposed, and the timing of exposure to the foreign antigen all influence this immunogenicity. So now let's move on to antibodies. Antibodies are specific proteins referred to as immunoglobulins. Many antibodies can be isolated in the gamma-globulin fraction of protein electrophoresis separation. So the term immunoglobulin, also referred to as Ig, has replaced gamma globulin because not all antibodies actually have the gamma electrophoretic mobility. So antibodies can be found in blood plasma, and in many of the body fluids like the tears, saliva, and colostrum. And that's just the first part of breast milk before the breast milk starts filling in with the fat solids. And that's the part that has all of the antibodies that the baby will need. So the primary function of an antibody in a body's defense is to actually combine with the antigen, which may be enough to actually neutralize the bacterial toxins or some of the viruses. Now, secondary interaction of an antibody molecule with another effector agent, something like complement, is usually required in order to dispose of the large antigens like your bacteria. So when we actually determine the Ig concentration, it can be very diagnostic for various infections and autoimmune diseases. Now, there are 5 distinct classes of immunoglobulin molecules that are recognized for most of the higher mammals, and these are IgM, IgG, IgA, IgD, and IgE. Now, the immunoglobulin classes can differ from each other in their characteristics such as the molecular weight or the serum concentration as well as functions. Now, the serum levels of immunoglobulins, which are IgM, IgG, and IgA, can vary by the demographic factors. So this would be like age and gender, as well as common habits like alcohol consumption and smoking, and any sort of metabolic abnormality like obesity um, or high triglycerides. So values are actually reported in milligrams per deciliter or international units. So let's further discuss the immunoglobulin classes and what each of them is responsible for. So IgM is the largest immunoglobulin molecule, and it is the first antibody that is actually produced in response to an antigen. It's a very large molecule, and because of that, it is confined to the blood. And so it also is very effective at agglutination and cytolytic type reactions. And in humans, it's found in smaller concentrations. It can be increased in various conditions like infectious diseases, like a bacterial infection, infectious mononucleosis, malaria. It can also be seen in collagen disorders as well as hematological disorders. IgG is the most abundant immunoglobulin in serum, and it is responsible for protection against viruses and bacteria. And it actually will diffuse more readily than some of the other immunoglobulins into those extravascular spaces. And there it can actually neutralize toxins and bind to the microorganisms. It is able to cross the placenta, and when there are actually IgG complexes that form, complement is activated. IgG does account for about 70 to 75% of the total IgG pool. IgG does have a very long half-life, so this kind of aids in its ability to actually be able to bind to specific Fc receptors. One of these most notably is the neonatal Fc receptor. So what this receptor does is it actually transports IgG from maternal circulation across the placenta barrier, and then it will actually transfer those maternal IgG antibodies across into the intestine within the neonate. Now, once it's there, this receptor will actually transport that IgG molecule across those cell walls without targeting any of the lysosomes that are there. It will then actually recycle to the cell surface, and then it will release IgG into circulation. So the fact that it actually has that intracellular sequestration of that IgG away from those lysosomes, it actually prevents it from being degraded as rapidly as some of the other serum proteins, and ultimately it allows those maternal IgG antibodies to actually be in circulation for that infant, so that once they are born, then they have those natural antibodies that were provided by the mother for the first few months of life. Decreased levels of IgG can actually be seen in primary deficiencies or genetic deficiencies, as well as secondary IgG deficiencies. If we see significant increases in IgG, this can be due to infectious diseases, collagen disorders, and other hematological disorders as well. IgA is the second most abundant immunoglobulin in serum, and it is predominant in the secretions such as tears, saliva, the colostrum, milk, and in intestinal fluids. It is synthesized largely by plasma cells that are actually located on the body's surfaces. If it's produced by cells in the intestinal wall, IgA can pass directly into the intestinal lumen, and then it can actually diffuse into the blood circulation. As it is transported through the intestinal epithelial cells or through the hepatocytes, it actually binds to a glycoprotein that is called a secretory component. This actually prevents it from being digested by the gastrointestinal proteolytic enzymes. And it does this by forming a complex molecule that we call a secretory IgA. And it is critical because it actually protects body surfaces against invading microorganisms, and because of this, it's able to be present in those mucosal type secretions like your tears and your saliva. IgA is decreased in primary and secondary IgG deficiencies, and it's increased in some concentrations An increase in IgA concentrations can be associated with infectious diseases like tuberculosis, collagen disorders such as rheumatoid arthritis, hematological disorders, as well as liver disease. IgD is found in very low concentrations in the plasma, and it accounts for about 1% of the total immunoglobulin pool. It is extremely susceptible to proteolytic or proteolysis, and is primarily a cell membrane immunoglobulin that's found on the surface of B lymphocytes, and it is associated with IgM. IgE is a trace plasma protein that is found in the blood plasma of unparasitized individuals, so patients who have not had any sort of parasite or don't have a parasite. IgE is critical because it actually mediates some types of hypersensitivity reactions or allergic reactions, as well as anaphylaxis. And it is generally responsible for an individual's immunity to invading parasites. The IgE molecule is unique in that it binds strongly to receptors on mast cells and basophils, and together with the antigen, it actually mediates the release of histamines and heparin from those cells. So now let's discuss a little bit about the antibody structure. So antibodies actually exhibit diversity among the different classes, which suggests that they perform different functions in addition to their primary function of antigen binding. Essentially, each immunoglobulin molecule is bifunctional. So you have one region of the molecule that involves the binding to the antigen, where you actually have a different region that mediates binding of the immunoglobulin to the host tissue, including the cells of the immune system and the first component of the classic complement system. The primary core of an antibody consists of the sequence of amino acid residues that are linked by the peptide bonds. So all antibodies have a common basic polypeptide structure with a three-dimensional configuration. The polypeptide chains are linked by covalent and non-covalent bonds, which actually produce a unit that is composed of a four-chain structure that's based on pairs of identical heavy and light chains. As you can see in the images depicted here, the IgG, IgD, and IgE only occur as a monomer of the four-chain unit, whereas IgA will actually have both a monomeric and a polymeric form. So you can see it there where it's almost like two that are linked together. And then IgM is seen as a pentamer, where you have 5 of the 4-chain subunits that are actually linked together. As you can see in this top image, a typical structure is arranged with 2 heavy chains and then as well as 2 light chains. So the heavy chains are represented by the blue, with the light chains represented as the pink. Then these are all linked together by disulfide bonds. And so the antigen binding portion of the molecule, which is the N terminal end, it actually shows heterogeneity that is known as a variable region. The rest of it is composed of relatively consistent amino acid sequences, and so this is considered the constant region. So here we have a nice chart that just kind of compares each of these different immunoglobulin classes so that you can see the actual structure of it, as well as look at other things like the molecular weight, the percentages within serum, whether or not it crosses the placenta, and its function. And you'll want to make sure that you're familiar with at least the structure of it, as well as whether or not it has the ability to cross the placenta, as well as their functions. And we will kind of get into a little bit more depth with these different antibodies and the structures once we start into our blood bank course. When an antigen is initially encountered, the cells of the immune system will recognize the antigen as non-self, and then they will either elicit an immune response or they'll become tolerant of it. It just depends on the circumstances. So an immune reaction can take the form of cell-mediated immunity, and this is immunity dependent on T cells and macrophages, or it may involve the production of antibodies. These antibodies will be as a result of your B lymphocytes and your plasma cells, and they are directed against that specific antigen. When the host lymphocytes come in contact with a foreign antigenic substance that binds to its receptors, then the host will actually start the production of antibodies. This will actually trigger activation and proliferation, or what we refer to as clonal selection. Clonal expansion of lymphocytes in response to an infection is necessary for an effective immune response. However, it requires 3 to 5 days for a sufficient number of clones to be produced and to actually differentiate into those antibody-producing types of cells. So unfortunately, this does allow a little bit of time for those pathogens to actually damage some of that host tissue and cells. So this is what we refer to as a primary antibody response. And with this particular response, an IgM antibody response takes place, and then that actually proceeds through 4 phases: a lag phase, a log phase, a plateau phase, and then a decline. Now, if there is subsequent exposure to the same antigen that produces a memory response, then you will actually see that because it reflects the outcome of the initial challenge. So in a case of antibody production, the quantity of IgM and IgG will vary. But when you have that secondary response, antibody response, this is because there's a subsequent exposure to the same antigenic stimuli that was once exposed to, so those antibodies are already kind of programmed into the system to produce those IgG type of antibodies. Now that secondary response will have the same phases as a primary response, but it differs in the fact that the timing of these types of antibodies as far as their production and the titer are a little bit different. So although the duration and the levels of antibody titers are dependent upon the characteristics of the antigen and the individual themselves, an IgM antibody response proceeds through 4 phases after there's actually an exposure to some sort of foreign antigen. So again, you have that lag phase. So you can see here in this image where it just kind of describes what's taking place as there's some sort of introduction, and then you have these antibodies that start to produce. So in your lag phase, you can see that all of them are pretty much kind of the same. You've got this phase where there's no antibody that's actually detected. And then, of course, you get that exposure and you have all of the response from your immune system, and then you go into the log phase. So you can see as you kind of come up with no detection, and then you start to actually have those antibody titers increase. And then you have a plateau phase where it just kind of peaks out. So you can see from your IgM, which is here labeled in blue, where it just kind of comes up and it peaks to a point, and then you actually have, once you've reached that plateau phase, then it will actually start to go into a decline phase, and so then the antibody starts to dissipate, and then you can see where it just kind of eventually works its way out. Now, if there is some sort of secondary stimuli that takes place, then you're going to see this response take place the same way. It's just going to take place at a different interval, as well as it's going to take place more rapidly. So you can see here that there's a subsequent exposure, and then you have that IgM that takes place. So whereas in your primary response you had that IgM take place first, and then as it starts to decline, you start to see that IgG start to peak up, and then that declines as well. With your secondary exposure, because your body already knows what it's supposed to do, then you'll see that IgM come into play, but then you'll also start to see that IgG come up a little bit sooner, and of course it's going to plateau and stay plateaued a little bit longer before it actually starts to decline. The ability of a particular antibody to combine with a particular antigen is referred to as its specificity. So this actually takes place in the Fab portion of the molecule, and it's known as the combining site. So evidence does suggest that an antigen may bind to a larger or even a separate part of this variable region. Now, the closer the fit is between this particular site and the antigen determinant, then that means you're going to have stronger non-covalent forces between them, and you're also going to have a higher affinity between the antigen and the antibody. Now, binding will depend on a close three-dimensional fit, which allows weak intermolecular forces to actually overcome the normal repulsion that exists between those molecules. So obviously, when you have more than one combining site that interacts with the same antigen, then that bond is actually going to be greatly increased in strength. Now, occasionally you do have some antigenic determinants that are similar on the various surfaces, so it's very possible that you can have a portion of the antibodies directed against a type of an antigen that is of a different type of an antigen. And this is called cross-reactivity. So you can actually see this in a lot of testing methods where you're using antibody type of testing techniques, and you actually have that antibody that will form some sort of bond to an antigen that it's not really supposed to be targeted at, and it's just because it's picking up a small portion of that FAB portion. Now, an example of this that we'll actually talk about once we start entering into our blood bank courses is that there's some cross-reactivity between bacteria that have the same cell wall polysaccharides as your erythrocytes, and so it affects the intestinal bacteria and blood typing, but we'll talk about this a little bit more once we get into blood bank. But that's just an example of cross-reactivity. Affinity is the initial force of attraction that exists between a single FAB site on an antibody molecule and a single epitope or determinant site on the corresponding antigen. The strength of the sum total of non-covalent interactions between a single antigen binding site and on an antibody and a single epitope is the affinity of the antibody for that particular epitope. Now, a low affinity antibody bonds, they actually are very weak and they tend to disassociate pretty easily, whereas high affinity antibodies, these bonds are more tightly bound, and so they tend to stay together a little bit longer. Antibodies that are produced in the late primary response usually have a higher affinity for an antigen than the antibodies that are actually produced early in the primary response. Now, the total functioning strength of all interactions between an antibody and its antigen is called the avidity. Now, when you have multivalent antigens that will combine with more than one of an antibody's combining sites, then the strength of the bonding is significantly increased. So in order for all of these antigen and antibodies to actually disassociate, then you would need all of the antigen-antibody bonds to actually be broken at the same time. The non-covalent combination of an antigen with its respective specific antibody is called an immune complex. Now, an immune complex may be of small or soluble, or it could be a large type, which is a precipitating type, and it just depends on the nature and the proportion of antigen and antibody. Now, all of this kind of helps with the basic principles of understanding some of these testing techniques. So it allows us the fact that we have antigens and antibodies that will attach to each other. We're able to see these agglutination type of reactions, and so we can see them through our testing methodologies. And so what we can actually do is mix a known and an unknown. So we would have a particular antigen or antibody that we know of, and we'll mix it with the opposite antigen or antibody, which would be like our unknown or our test patient, and we're looking for these reactions. So it could be like precipitation type reactions, hemolysis testing, enzyme linked immunoassays. So there are a wide variety of test methods that actually use antigen antibody types of reactions. There are a couple other antibodies that are antibody types that I like to make mention of, and these are neutralizing antibodies and monoclonal antibodies. So neutralizing antibodies are a subset of antibodies that are important in a specific defense against a viral invader. So neutralizing antibodies actually bind to a virus, and they bind in a way that it actually blocks the infection. So during viral infections, the human immune system will develop these neutralizing antibodies, as well as broadly neutralizing antibodies, and then non-neutralizing antibodies. Neutralizing antibodies are a hallmark of infections that are associated with various viruses, but in contrast, only 10 to 30% of infected individuals actually develop the broadly neutralizing antibodies, and that will actually show a broad neutralization activity. We will discuss a little bit more about what monoclonal antibodies are, but just for the time being, just understand that they are purified, cloned antibodies that are from a single cell source. There's a lot of current studies out there on these, and as far as using them for various testing methods and looking for various diseases. But we're not really going to focus on that right now. We'll look at this a little bit more once we actually dive into our blood bank courses.